Reviewing.
Are you going to just chi-squared or?
No. Really, statistics. Chi-squared counts as good, but yeah.
I remember not liking it at all.
That's about my position. So we're on here. For those who are not yet aware, I have been asked to teach a brewing course. It'll be a 3-credit course. It'll be a year from now. It'll be in the spring. So a year from now, I'll be teaching the brewing course. 2 years from now, I will not be teaching it. I'll be teaching botany. 3 years from now, I'll be teaching the brewing course. So it's every other year. So if you're a junior, your only chance to take it will be in the spring. If you're a sophomore, you get, I guess as a sophomore, you take it your junior year, and then your senior year, it won't be offered, right? So basically, you got one shot at it. I think I'm going to have to offer multiple sections. No.
If you were like a chemical engineer, would that be counting as your chemical engineering elective?
Yeah, it is a CPEN course. It is CPN 398.
Would that be biochem or biochemical?
It counts as a bio, I think it's a biochemical. I don't know the details on it yet.
Come back.
So how many people, in general, how many people would be interested in taking that course a year from now?
I won't.
I'll tell you a little bit about this before your people ask questions.
You're closing already.
This is the process. The farmer grows up barley. They sell raw barley to the maltster. The maltster germinates the barley. That's called malting. The germinated barley is then dried, sold to a brewer who incubates it in water to get a sugar solution, and then they ferment that into beer. They package it. They gives it to a distributor who then, does all the sales and marketing stuff. I teach barley growing in my botany course right here. And last summer I had a really cool opportunity to go to barley field school. Did I tell you guys about this? What I did last summer? There's a guy in North Dakota who has a breeding program for barley. And the idea is, we've selected for these barleys that are really great for brewers. And they've found barley they've done this worldwide search for different wild variants of barley. They found one in Ethiopia, which is the genetic profile of that is so different from the bred barley that we have here that we're trying to breed characteristics in. They get a more robust barley that rich barley that we grow for our beers and such. So this guy runs 16,000 crosses of barleys every year, and then he grows it up in North Dakota, and then they grow it in Arizona, and they grow it in Australia or South America or something like this. And it's 3 crops each year, so 3 generations each year instead of 1. And some areas he grows it so then it's susceptible to getting exposed to certain barley diseases. And anything that dies off, well, we don't want that anyway. And he grows it like in Arizona, and he doesn't water it, so anything that's not drought tolerant dies off, and you want those anyway. Every year it goes on, so for 10 years, so 30 generations, he selects down, he gets down to like 1 to 3 different things that survive. And so he grows up a field of that, and he sends it to a maltster, they malt it, and they send that off to Anheuser-Busch, and Anheuser-Busch brews a beer with it. And every year for the 30 years of this guy's career, Anheuser-Busch has said, we don't like the flavor, throw it away. So imagine all the work the guy does. So when I heard about this, I was like, well, Anheuser-Busch, the product that Anheuser-Busch makes in general, so a craft brewer is trying to make things that have flavor that appeal to me. Anheuser-Busch is trying to make a product that does not offend anyone so that everybody can drink it. So what Anheuser-Busch considers a good flavor in beer is not what a craft brewer would consider a good flavor in beer. So what I want to do is I want to short circuit that system. When he gets his crosses down to about 400, so every year he'll have 400 coming through. I'm going to take those and I'm going to malt those in a pilot small malting system that Epix Group is actually putting together for me. Apex 2 group is designing it. It'll be built this summer. We're going to brew 400 different batches of beer, 4 bottles of beer. And then I have contacts and craft brewers here in the metro area. They will all come in and have a tasting panel. And with the 3 bottles of the beer, they will drink those and decide, do I like the flavor of this grain or not? Because what they're looking for flavor is different from what Anheuser-Busch is. And then the head brewer in Belgium actually called me and said, wow, I'm really interested. Can we get some of those beers too? So probably one beer will go to New Belgium for their testing. The idea is we can short circuit the system. Things that may have been dumped by Anheuser-Busch, we can pull those out and do a flavor characterization, not just drought tolerant, not just disease resistant, but tastes good. And I think we can get an amazing revolution of barley flavors in beers. So we will focus a lot on the malting process in this. We have a course called Bioprocess Engineering where you can already learn about the brewing process. We'll go over that, but we'll look at it more like process control stuff. Your beer is oxidized. How can you design your system and test to find where the oxidation occurred, where the contamination occurred, or whatever like that. So that's the longer than I wanted to talk about that. Today we're doing a review session because we have an exam on Monday. So if you guys have any questions or want to ask me anything, now is the time to do it. I have not quite finished the grading. The dihydrogen cross field was difficult to grade, so I can get through all of that and finish that up before I leave today. And what I'll do is I'll put the worksheets, I'll just put those in the box outside my office and go and pick those up. Okay, anyone have questions? Can I talk about complementary gene action? Yeah. So we have a biochemical pathway. This chemical has no color. This chemical has no color. This chemical has blue. So I can have a flower, and this flower is white, because this flower has a mutation and can't perform this step. So we never get N, and we never get O, because we can never make N, right? We have another flower which is also white. This guy can make N, but it can't make O. Make sense?
Yeah.
If you gave this guy, if you injected chemical N into this guy, right here, it could finish the process and it would actually be a blue flower. Right? So we can say that this guy is, let's give this gene, we'll call it gene A, we'll call it gene B, these are the enzymes. So this guy is defective A, but it's functional B and functional B. And this guy over here is functional A and functional A, but it doesn't have a functional gene B. So the effect, the function of one gene can hide or mask the function of another gene. If we were to cross these guys together, we have a functional gene B and a functional gene A, which means we complete the pathway. Both of these work. You take a white flower and a white flower, you cross them together, and you get blue flowers. You have a pure breeding white and a pure breeding white, and you cross them together and you get blue flowers. Does that make sense? We understand the logic behind it. When we talk about this in terms of biochemical pathways, I was introducing to you the concept of complementary gene action. We will discuss complementation assays when we get to the cell cycle, which is the cancer part of the course. This is the fundamental understanding you need to do that in great detail in these types of experiments. Did that help? Does that answer your questions?
Oh, yes.
Thank you.
It is.
These genes are complementary to each other, A and B.
Oh.
Can you guys work through a 3-point cross?
Yes.
There is definitely a question. I'm going to make up numbers and put it together. I have no idea what the arrangement of the genes is and how far apart they are. I guarantee that is on the exam. It'll be exactly like the worksheet. I'll probably take the worksheet, copy and paste, and then just change the numbers.
For a 3-point cross, is the parent generation, is it always like homozygous, like big A, big A, big A? Or can it ever be big
Yeah.
A?
Oh, okay, so your question is, right,
Yeah, you can be.
crossed with, oops. So that's the cross, and your question is, could I ever get something like this? Right? Or this is a big D here like this? The answer is no, because this is not pure breeding.
Okay.
You always, always, always come from pure breeding strains.
So that way then, like in our test cross, the baby of that will always be heterozygous.
Yeah, so you always get a heterozygote out of this. And then you do your test cross. And test cross means what? Homozygous recessive at all. Homozygous recessive at all. Other questions? You're the only one prepared? Roll them all up.
Okay, so it talked about like with the storage, you should identify types of crosses. This is like one of your goals, and then it talks about like which ones violate or follow Mendel's 2 laws. Do multiple alleles follow Mendel's laws?
Yes.
Do they all follow? What like and like epistatic?
Those do not follow Mendel's laws. Organelle inheritance, maternal effect. So probably the way the exam is going to be written is probably a very straightforward Mendel type crosses are early on, and then I ask you the bulk of the rest of the parts of the course, and then I get into the really screwy types of examples, and then I hit you with the problems and the long answers. That's probably how it would be organized.
So is there any way you can just like straight up be like these don't follow Mendel's 2 laws? Organelle inheritance?
I think I'll say you run this cross and the data is this. It doesn't look like it followed Mendel's laws. Which law is violated or which law is followed? Does that make sense?
Yeah.
There's a law of segregation law and sort of kind of same thing, just one is for one characteristic and the other is for 2.
No, the law of segregation is that an organism has 2 copies of the factor, right? 2 copies of the code, and those segregate independently. And then, so you get gametes formed in the male and gametes formed in the female. And according to Mendel, they find each other randomly, so you get random mixing. That's for segregation. For independent assortment is, you've got that going, you've got the segregation going on, but in addition, if you have a gene for blue eyes and genes for hitchhiker's thumb versus non-hitchhiker's thumb, the allele for that codes for blue eye is not connected to the allele for this. They're on different chromosomes, right? Because Mendel didn't know that there were different chromosomes. And you know what? Mendel followed 7 traits. 2 of the traits he followed are on the same chromosome. They appeared to assort independently. Why did they, if they're on the same chromosome, how could they possibly have appeared to assort independently? Crossing over. They are so far apart on the same chromosome that they crossed over so frequently that they appeared to segregate independently. Makes sense? Did I just confuse you with that example? Okay. He got lucky. If they'd been close, he would have come up with this model and then had this example that destroyed it. He would have been, I think he wouldn't have concluded anything, right? Or maybe he would have just like squashed those organisms and something.
Which laws can be proven with the monohybrid and which need the dihybrid?
Segregation, independent assortment. The way I remembered it when I was in your shoes is the cheating. I'd like you to conceptually know what one is and conceptually know what the other is and then figure it out from that. But the way I did it when I was in your shoes is I cheated and I said, this is one word, mono. This is 2 words, di. The way I learned it is I had that little mnemonic, and then on the exam I go, okay, if I have a dihybrid cross, what's that about? Well, it's gene A and gene B, so that it shows gene A segregates independently of gene B, independent assortment. I like sharing all these little mnemonics. Actually, most of them I've forgotten. Because yeah, apparently they were, or I finally learned it after 8 years of teaching it here in class. So I don't need a mnemonic anymore. Does anyone else have mnemonics that they want to share? You haven't studied for the exam yet, so you don't know what your mnemonics are. I know how it goes. Yeah.
So does that mean that you can prove blending or unequal distribution from either cross?
Yeah, from a monohybrid you can demonstrate the law of blending fails because you just have to go to the F2, right? And you'll get some of the originals out. Even if you have red and white, you get pink as an F1. If you self-cross those for the F2, you can get red, white, and pink out of both, right? And then for equal contribution, you can do a monohybrid, but you have to do the reciprocal cross. If you have red and white, or red female, white male, you have to then switch it so the female is white and the red is male, and you have to do a second monohybrid cross. Law of equal contribution.
Okay.
Or if you're trying to show organelle inheritance, the law of unequal contribution. Did you have one? You raised your hand at one point.
Yeah, are you going to have us do any of like the wild, like as you briefly mentioned like wild type prevalence and how you can have like more than one wild type with like a certain percentage in the population? Or you have to know like what population?
You don't have to know, you get to know. Okay. Do you get to demonstrate mastery of understanding that? So when I use wild type, general innocence is wild type, we would say brown eyes are wild type. And quite often people are using that term when they mean dominant. And if you leave this classroom, you go out in the world, and someone says, oh, the wild type color in human population is brown. You know, the dominant allele is brown. geneticists, for whatever reason, came up with a definition. If an allele exists at 5% of the population or more, it's called wild type. So in humans, blue eyes as an allele is wild type. It's recessive to brown. Brown is also wild type. Does that make sense? So I'm not too picky about you have to learn and memorize. But understand that you can have an allele that's much more prominent. And you can have two alleles that are very dominant in the population. And in some cases, you can have 4 alleles that are all wild type because they appear in the population more than 5% each. You don't have to do anything. You're welcome to miss that question in the exam. It's always an opportunity.
Can we talk about recombinant or recombinant? Is it word vocabulary word?
Recombinant?
Yeah.
Recombinant means the genetics have gotten all mixed up. There's 2 ways that recombination is used. In genetic recombination, that happens through sexual reproduction, the chromosomes get all mixed up and segregate independently during meiosis and then recombine, and so the progeny have a definite genetic profile that's different than either parent. That's recombination through sexual reproduction. And then we learned about recombination, and we talked about the actual physical exchange of DNA between chromosomes. The crossing over is a genetic recombination of itself, a different way to use that same word.
How much in detail should we know about like the different phases of prophase?
In Prophase 1, in meiosis, you should know where crossing over occurs. You should recognize the phase names. So in mitosis, we break it down in prophase, metaphase, anaphase, telophase, meiosis, right? In meiosis, because there's that special crossing over event that happens in Prophase 1, the people who study crossing over have broken Prophase 1 down into all its smaller little steps, and leptopene and all these other specifics. And just to learn about specific, primarily when does the crossing over occur, and more conceptually, what is happening in the crossing over. If you need to have conceptually what's going on, because when we talk about DNA replication later in the course, we will talk about how the crossing over actually occurs. We'll talk about the holiday junction and these types of things. We'll show physically how it happens. And, in the past, I've done it all on computer, just PowerPoint files and showing how things move around, how chromosome pieces move all around. And definitely students didn't get it. So I need to create a model in class to show how this stuff gets all mixed around and how things get broken and pieced together. I'm not entirely sure how I'm going to do that. Probably just some tubing with connections. It's pretty cool when you see how it all happens.
Can you give an example of codominance and incomplete dominance?
So incomplete dominance is an example, is the flower example, where you have a pure breeding and a pure breeding, and you mix them together, these pure breedings, so that you've got the heterozygote, and it doesn't look like either parent. It's kind of in between the two. So if you had brown eyes and blue eyes, and you breed them together, and you get like a crappy dark gray. It's like blue and brown mixed together. It's kind of like my eyes, right? And they're like steel, my wife calls them steel blue, cold steel blue eyes, she says. You mix them together and you get something in between. That's incomplete dominant. Red is incompletely dominant to white, you get pink. Co-dominant is you cross together and you look like both parents at the exact same time. So if you have red flowers and white flowers and you cross them together, you get flowers, and there's like a red stripe and a white stripe and a red stripe and a white stripe. Does that help?
Yeah.
Blood type is probably the example to study for codominance. Shannon, did you have one?
Yeah, could you talk a little bit about physical markers and genetic markers?
Yeah, a physical marker is if you have a chromosome. And I've drawn this chromosome to be a little smaller than this one on purpose. A physical marker, so we stain with this Meiosis stain. We did the banding pattern on here. So all of these bands on here are physical markers. Right? So this guy is physically, if you look under a microscope, physically it looks different than this guy because this part of the chromosome is missing. Right? That band is physically missing. What we talked about in an example when we were in class is we were talking about there was someone found a chromosome where there's some other piece of some other chromosome hanging off the edge there. You look in a microscope, you can physically see the chromosome is different than this guy over here, right? A genetic marker is a trait. Brown eyes, blue eyes, red flower, white flower. Small wings, miniature wings, or normal wild type normal wings, right? So far, I've got 100%. No one's stumped me yet.
Can you answer this question on lecture 12? It says, explain how to determine the default pathway of sexual development in a newly discovered species.
What is the default pathway for sexual development in a newly discovered species? Yeah. So let's use humans as an example. Do humans develop as male or female as the? So if you've got a human, it's going to develop as to either male or female. How do you deviate off that to get the other one? Is this female and male is like forcing the system into something different? Or is this male and the females are forced off to a different pathway? You understand what I'm talking about? Anyone want to take a guess as to how you would do that?
Default is female.
Why?
Because you have to add testosterone to push development of the male to the type.
So how could you show that? That's the question I'm
How
asking.
do you show it?
What would be the experiment you run?
Remove testosterone from the system.
So you could have an XY and somehow pull the testosterone out as it's being produced and see if this develops male or female. Okay, that's one way. So, I mean, fundamentally the question is that female because 2X says female? Or is this male because a Y says male?
So to the XXY to come out.
So if a second X says you're going to be female, this should be a female, right? That's another way of doing it. I think your way works. It's harder to do, but this way, you just screen a whole bunch of males and see if you can ever find an XXY. In humans, this is called Klinefelter Syndrome. And that is a male. So people, it looks like in humans, the reason people are female is that the Y directs off of the default female pathway. Makes sense? Klinefelter syndrome, normal fellas, physical feature, they often have a depression in their chest right here. It's a pit that kind of goes down in their chest like this. I can remember there was a screening at my school, and one of the kids on my soccer team said, you know, he learned, wait, a depression, I've got one right here. He looked at me, I got a depression in my chest right here. And he called him out of class, and I'm not sure I ever saw him come back. You know, he just kind of like flipped out. So he was probably an XXY. That happens. So mentally, these people are. I think the IQ is maybe a little bit lower, but I mean they're not all going to be like really dumb, you know, like handicapped and severely handicapped. So there's probably in a population of 4,000 students here, there's got to be a couple of these fellows on this campus. A girl I knew in college was XY. Did I tell you about this? And what happened is the genes on the Y that control this break off to become male, she had a mutation in one of those genes such that the pathway was incomplete. So she, by default, developed as a female. But she wasn't fully intact as a female, apparently. Her internal parts didn't develop female. She, on the outside, looked female. Just a good friend of mine. Someone who was in my little clique of friends. Never knew about it. So that happens.
How did you find that out?
When I was in graduate school, someone was talking about, I can't remember the name of that disease. And there was a picture, and it was Katie. It was a clinical picture, and it was my friend. I was like, holy shit.
And how did you figure it out? How did she figure that out?
Her period never started. She figured out when she was a teenager. Her period never started, so they were like, well, what's going on? And took a look on her eyes. They don't have the right parts in there. They didn't have any parts in there, and so then they did a genetic test. Yeah, she was sad that she'd never have kids. She'd adopt kids if she wants kids. But it didn't faze me at all. She's the same person. Okay, other questions? Now that we're in deep thought here. all the guys are like.
So since chickens are the opposite then, is it the same thing where if it's like a WWZ, does it develop as a female?
I don't know the answer to that for chickens. I would guess a WWZ would develop. I don't know the answer to that one. That's a great question for the exam. WWZ, so in chickens, WW is male, right? And WZ is female, right? That's how it works. So WWZ would be, you guess, would be male, right? Why would it be female? Because it's got a Z. Yeah, maybe it'd be female because the Z directs maybe the default pathway in chickens is male. I don't know the answer to that. Because it could be having two W's means you're going to be male, in case this is male. Or it could be genes on here direct being female, and it doesn't matter how many W's you have, you're going to be female. That's the fundamental question. I don't know the answer to it, sorry.
But like that's how you'd set up the question on the test, would be like,
Yeah.
all right.
For those of you who would be preparing for the MCAT, expect them to say, you have a sex-linked disease, and it's usually barred feathers, and the comb is whatever. There's many different kinds of comb. On the MCAT, they will ask that question. You have a sex-linked trait in chickens, and here's your results, explain it. And you just have to know that these guys are backwards. And they'll have a whole paragraph on the last year, like 3 to 5 questions. Definitely expect that at the MCAT. Okay, you got me. I'm down to 95%. I'm just not going to say 95%. If that was a small percent of the exam.
Because chicken genetics is totally going to be on the exam.
We play a game, I play a game this year, a little commercial break while you try and come up with more questions. Driving down the road with my girls, we play this game of who am I? I'm a character from Toy Story. Who am I? You start guessing. It's all fun. I'm an animal that's pink. Who am I? A pig. No. Flamingo. Yeah. Oh, great. So it came to my turn. I was totally running out of things, and I said, I'm an animal that's really tasty. Chicken, pig, a goat, sheep. They're rattling all these out. Beef. Close. Where do you get beef? Cow. Yeah, cow. Pig. My kids. I don't know if I told you this. Also, this week, Kate's like, Daddy, do frogs have blood? Yeah. Do pigs have blood? Yep, pigs have blood. Do mosquitoes have blood? Not quite. It's called hemolymph. She goes, hemolymph, okay. She's 5, you know, she's totally gonna nerd out. Worse than even me. Your commercial break there.
Just trying to get it all sort of laid out. There's going to be like a Mendel section where it's just
Story problems, pretty straightforward stuff.
And then it's going to get just sort of slightly more.
And then I'll give the bulk of the test, which is not those kinds of story problems.
Yeah.
And then at the end will be the challenging, tricky ones and stuff.
Yeah. Like sort of one per weird concept kind of deal.
Yeah. If you go to the key points, I will try to write a multiple choice question for every key point. They will be in that order on the exam. The story problems, I'll probably mix them up a little bit. Those, there might be 5 of those, and then mix those around.
For the worksheet, when we were crossing two things, and like your examples in class for determining the likelihood of a genotype or a phenotype were just from like one, like the offspring. Or I don't, how do you,
So you're talking about I was doing like this cross kind of
I'm
thing,
very good at
right?
Yeah, but you said it was like the possibility of it being either parent. Like how does the word either affect the way you're going to do the
Yeah, so let's do this one. Let's figure this out. What are the odds? And it'll look like, what are the odds that it will be this?
Look like or have that exact genetic?
Say genotypically be this.
Okay.
So I need to do a Punnett square for every one of these. So this one, that's the Punnett square. This guy can only give a big A. This guy can only give a little a. 1 out of 1. Right? So this guy, oh, it turns out it's the same thing. So 1 out of 1. This guy is, you see how I'm doing this? Oops. So that's 1 out of 2, right? D is going to be 1 out of 2. E is going to be 1 out of 2, is that right? Okay, so that is 1 out of 8 then. So I've changed it only by this guy. What if I say this, and it could also, or this one here. So this is going to be 1 out of 1, 1 out of 1, 1 out of 2, 1 out of 2. What's that, 1 out of 2? So this is also 1 out of 8. So the odds of it being this guy or this guy is 1 out of 8 plus 1 out of 8. But you add them together. On the worksheet, a lot of you missed one of those because the cross was this. By. It's the only time I've ever tried to trick you guys. This parent looks like this parent, but genetically they're identical. So if you say, oh, the odds of this is 1 over 16 and the odds of this are 1 over 16, right, 1 over 16, then it's a 2 out of 16 chance. Nope, because this parent looks like this. So there's only 1 in 16 chance it looked like the parent. Isn't that tricky? How many of you got it? Not many. You understand what the trick on it? You're looking at me like you don't get it, Molly.
Also kind of sleepy.
Yeah. No, we're doing just something. So if the parents were that, say, okay, this parent looks different than this, so yeah, you add them together. But this is the exact same parent as that one. So if it looks like this one, it has to look like this one. So if it looks like this one, you can't add anything else here. Normally I don't trick you guys like that. I consider that a trick question. I think it's unfair.
Well, when your questions were looks like,
Yeah, that'd be phenotype. So that, yeah, it looks like, then you'd say, oh, well, you know, then you're, well, actually, looks like would be like this. I don't care what that is. Well, that'd be the same. I don't care what this is. I don't care what this is. Right? That'd be phenotype.
How much of like the imprinting and the exist and all that stuff when it's forming the bar body are we going to know since we kind of skimmed over it pretty quickly?
Yeah. I'm not sure I'll even have a question on there. On there, it'll just basically say, so I didn't say in class, but remember when we were talking about expressivity, penetrance? How can you have someone who's pure breeding brown, someone who's pure breeding blue, crossing together and get blue eyes if we know that brown is dominant over blue? How could that possibly happen? And it can be that you genetically should be brown-eyed, but that brown eye allele is not expressed because it gets packaged away somehow through one of these epigenetic methods. So the DNA gets methylated and then you can't express that gene.
Okay.
The only thing you express is the blue, that's it. That's why you can have a difference in your genotype and what you actually express. So that really throws a wrench into Mendel. Some of this stuff, so this wave of obesity that's sweeping over the US, people who are eating a certain diet and living a very sedate life, if you don't use genes, your body methylates the DNA to get rid of it because you're not using it. And that stuff gets locked and packaged away. So once a person stops exercising and becomes unfit, it's very difficult to get those genes back out, and the person can never become a fit athlete again. And the scary thing is, the methyl groups that are put on the DNA, when a parent is going to pass some inheritance pattern, epigenetic inheritance pattern down from itself to its progeny, it uses methylation of DNA. So if you are methylating your DNA and locking it away, then you can't use it and you're going to be unfit. You can pass that methylated pattern on to your children, so that those genes are unavailable to your children, that your children will be fat and unfit because you were a lazy ass. So I see this wave of obesity as a serious thing. It can be passed from potentially passed from generation to generation to generation. And the only way to break it is for someone to say, I'm fat and my whole family's fat, but I'm going to get off my ass and exercise and go through the hard effort of stripping that DNA out of its locked state. It's very hard to do. So the saying is, if you don't use it, you will lose it. It takes 7 years of continual effort to basically lock those genes. See you at the gym this afternoon. Okay. If you have other questions, I'll be in my office. I'll be leaving around 4:30.